Sheri, take it away.
Hello, AP Biology students and teachers. It's wonderful to have you this afternoon for a little review on natural selection and evolution. I am going to share a document in the chat. And students, if you could open up this document, this will help you to follow along in our lesson today. And teachers, in your email, you have a document as well that has all of the corresponding answer key and solutions to the work that we will do today. So without further ado, let me share my screen and we will get started. Let's see, I'm going to share my let me make this full screen for us. Okay, friends, let's make sure everyone can see this document. Are we good to go? You can give me a thumbs up. I can see some of your faces. All right, wonderful. Okay.
I have a question.
Yes.
How do you get to this worksheet?
Okay, if you look in your chat box, there should be a link there that you can click on. Are you able to see your chat box? Awesome. If you can see that, then you just open up that link and you'll see the doc there. Okay, are we good to go? Wonderful. Thank you for asking a question. We want you to do that. Okay, two of our engagement strategies that we will use today will be waterfall chat and launch a poll. So the first strategy, what you will do is you will post in the chat box a response, but you don't post it immediately. You think about it first, you type it in, but we wait and we post all at the same time. Okay, so this is, well, could be a bio-related question, but could not be. So I want you to think about something fun that you may do after you study today. Some of you have been studying all morning long with other AP Bio study sessions, and now you're here, you're awesome, you're ready to learn AP Biology and work on this for the next couple of hours. What's something fun that you're going to do after you study today? Think about it. Go ahead, type it in the chat, but do not press send until I say waterfall chat. Everybody have something in there? Okay, let's press send. Do we have some chats coming up there? I hope so. I hope you're sharing. I can't see it, Mr. Akampora, but are they sharing a chat?
I can only see, oh, we've got I'm throwing a baseball with my daughter. Someone's going to their sister's graduation dinner. Someone will be vegging out and watching TV shows. I hope they're classic TV shows, a little computer work. So looks like you guys need to get out more. It's a beautiful day here.
Okay, wonderful. So that's
okay, I'm putting on my parent hat. Get off the couch and get out there and get some exercise. All of you.
That's a wonderful opportunity for waterfall chat. Remember, don't share until I say waterfall chat and we all type in the chat box at the same time. Okay, and Lou, if you could kind of monitor those as we work.
I will be doing that, absolutely. I'm enjoying it.
Okay, so everyone can see our learning objectives. We have several objectives in this unit. In our 50 minutes together, we will not be able to cover everything, but I'm going to try to hit on what I call the top three: natural selection, populations in Hardy-Weinberg equilibrium, and phylogenetic trees. So those are really important concepts. So I want to make sure I touch on those, but I'll try to get in as much as I possibly can. So natural selection. I love this unit. It's one of my favorite. Actually, the math, Hardy-Weinberg equations are my favorite math in AP Biology. I also like chi-square really well, but I love a good Hardy-Weinberg. So what I want you to do, if you have this doc open, you have it in view mode, so you can't type on it, but you can file, make a copy of the document, and type in as we work together. So here's our first waterfall chat. Are we ready? Natural selection is one mechanism of what? The theory that populations don't respond yet, just type it in. The theory that populations that are better adapted to their environment will survive and what? You should have 2 words that you have chosen for our waterfall chat. So you should have them typed in. Are we ready? Let's waterfall chat. Are we responding? Oh, we have someone that needs in the weight room. So I guess I can admit them. Let me go ahead and admit them. Do we have responses, Mr.
We
Akinport?
do. I'm actually, since I don't know this material, I'm responding myself, but the students are saying evolution and reproduce.
Ooh, you
Seems
are
to be.
fine and correct. Okay, so let's see. I'm going to type in. Let me go here. I think it should let me type in. And I won't be, so that we will be able to move more quickly. I will not be typing in all of this because it will take a little bit of time. But that's how it's going to work today. Okay, so follow along. Some of the blanks I will give you, others you will do a waterfall chat with. Great job on that first one. Excellent, students. You got this. So that's pretty much the basic idea and concept of everything that we will be talking about with natural selection. I've chosen some images just from online to kind of further explore our thinking and the causes for natural selection. How does this happen? What causes this to occur? And we know that typically it's competition for limited resources. And that results in differential survival. Some organisms survive and others do not. And those individuals with more favorable phenotypes are more likely to survive and reproduce offspring. Therefore, they can pass those traits on to future generations. Now, one of my favorite activities in natural selection is rock pocket mouse. I love that activity from HHMI. And this is just a pictorial of something similar to what we have done with rock pocket mouse in the past. You're going to see here are some mice of 2 different shades of phenotype colors that are living on a black rock. The bird is preying upon some of the mice, and which ones will probably be preyed upon the most? Well, the ones that the bird can see the best and take in. So the next generation is going to have what? Fewer mice that were not preyed upon. So let's think about this next question down here. How do we measure evolutionary fitness? So what is so important about how we can measure evolutionary fitness? Think for just a moment and let's put a few thoughts into our chat about what we think. How do we measure this? So we're going to try our waterfall chat. And you've typed in a response. So waterfall chat.
Okay, now?
Yes, sir. What do we
Okay,
have?
sorry. I need just a minute more to type in there. Waiting on some responses. Come on, folks.
Come on, what do you think? It's really more simple, and we've already actually discussed this. How do we measure evolutionary fitness? Well, what's the most important thing to do for the next generation? It's by reproductive success. So let's type in reproductive success. Okay. Those organisms can pass on those wonderful traits that allow them to be the most successful for survival. So reproductive success is how we're going to measure evolutionary fitness. What about what affects the rate and direction of evolution? What do you think? What affects the rate and direction? of evolution? I'll give you this one, but I do want you to be thinking about it. So actually, it's the ability of what? Biotic and abiotic environments. So that's very important to this survival thinking. And different genetic variations can be selected in each generation. So just keep that in mind. I won't type all that in because we need to cover a lot of ground today, but think about that. Your teachers will have the solutions for each one of these questions, but I will repeat this if you're typing in this document. The ability of abiotic and biotic environments and different genetic variations can be selected in each generation. Awesome.
Ms.
Okay.
Bonzo, could you expand on that just a little bit further? What do you mean by a biotic and abiotic environments?
Good question. I love that. Okay, so let's think about bio. What does bio mean?
Living, I think.
Living. Excellent. Okay, so living environments versus what would be abiotic?
Not living. We're dead.
Yeah. And I'll often compare, certain environments, a rock. So a rock is something that could be considered what? Abiotic. So anything that's living or non-living, we use those terms often when we're talking about our environments. So Mr. Akampora, that was a wonderful question. Thank you.
Well, thank you.
All right, so let's move on. And if I'm going too quickly, please tell me, students. And can everyone see this document?
Yes.
Okay, perfect. All right. Well, does everyone have this document open? Your teachers have this and they can share it with you if you don't have it in front of you right now. But we also posted it into the chat. If you joined a little bit later, you may not have this in your chat box. So I might ask Mr. Akampora if he could copy and paste it in one more time, just in case.
I'm trying to. This may be beyond my, I'm trying. I'm not a computer person.
Okay, you're wonderful. Just let me.
I'm working on it. I'm working on it.
Fabulous. Okay, I'll keep moving on.
I think I got it.
Nice, Mr. Akampora. Thank you. Okay, describe the importance of phenotypic variation in a population. Describe the importance. Okay, so this is where we have to think about what natural selection acts on. So obviously, we kind of answered our own question there. It acts on what? Phenotypic variations. So I can type in phenotypic variations in populations as what? What's going to change? What did we just talk about a moment ago? Abiotic and biotic what? What's going to change? So let's have our waterfall chat so we can fill in this idea. You should have one word as what change. Selective pressures applied to populations. So what's going to change? Okay, waterfall chat. Let's respond. What should go right here in this blank? Do we have some responses?
We do.
Mr.
We have my bad response, but we also have some environments seems to be a very good response from two of our participants here.
Environment is perfection. Good job, guys. So you should have environments in there because that's what we just talked about, right? Abiotic and biotic environments. So wonderful. Okay, a couple more things. Let's see, phenotypic variations can significantly increase or decrease what of the organism in particular environments? Phenotypic variations can significantly increase or decrease what of the organism in particular environments? What is it that's so important that we were trying to measure? Think of what do you think would fit best there? Okay, you got your idea? type it in the chat. Waterfall chat. Anybody have this one?
I said fitness, we have natural selection, and then a few people who are stumbling on their keyboards because they should be typing in there. Come on, folks.
No worries.
Please join us here.
Perfect. Okay, so fitness fits wonderfully. Great job, guys. Okay, so how does the trait change? So how does the trait change? And what kind of natural selection? Typically, my students, we learn about 3 different kinds of natural selection: directional, stabilizing, and disruptive. And I love this little pictorial that I just find on the internet, and then I apply it. I have the source down here below, but this is just from Quizlet, which I use often in my classroom for little quizzes for my students. And in this type of natural selection, we are selecting against the other extreme. So you could see this beautiful organism. The one with the long tail is going to have an appearance of a snake. So it's actually going to frighten predators away. And you're going to have more of an advantage if you are this organism with a longer tail. So in comparison, stabilizing selection, we're going to have the most advantage in our population if we have a moderate trait. So here we have an example of a cat with a short tail or a cat with a long tail. And in this case, having the medium-sized tail is going to be the most advantageous for that population. And then again, disruptive selection. This should all be somewhat review for you. And we would know that we're going to have the most advantage if we have the extreme traits. So short tails in the squirrel help keep predators from catching you on the ground, but long tails are good for balance in the trees. Medium tails, well, they really don't help. So in just a moment, I want to get to Hardy-Weinberg, but we're going to practice launching a poll. I have a few more review topics here in our learning, and then we will look at an AP Bio question about natural selection. Let's see. Explain how humans can affect diversity within a population. Humans can affect variation in other species through, now this is not per se, we wouldn't say natural selection, but how do humans affect variation? What kind of selection do we call that? Let's have a waterfall chat. Are we ready? Okay, type in what you think. Don't hit send yet. Okay, here we go. Waterfall chat. Do we have some responses?
And we've got quite a few people saying artificial, including me. And I knew that, Ms. Bonzo, but will you give us maybe an example?
Oh, yes, sir. Absolutely. Okay. How about, let me see if a student has an example.
That would
And
be
then
great too.
if we don't get examples, I'll give you 2. Okay. All right. Let's see. Come on, kiddos. What's an example of artificial selection? Some of you that are farmers or in the farming industry, I know you're going to give me a good one. So go ahead and type it in the chat box and waterfall chat.
We have cattle and animal breeding.
Yes. Excellent. Anything else? I'm going to take my favorite.
Something specific would be terrific. I'm just trying to, I know dog breeding was probably in there, and I'll throw in a plug for the movie Best in Show.
You have chosen my favorite, Mr. Akampora. Dog breeding, Wow, we've been doing this for generations, haven't we? And selecting the most favorable traits for the type of beautiful dogs that we would like to have. So that's an excellent example of artificial selection. And I had some pictures that I was going to put in there as well, but my slide presentation was getting a little bit lengthy, so I didn't put that in there. But I think you can probably, if that is on your AP Bio exam as a possible question, I think you're going to rock it out. You're going to do great on that one, so no worries. Okay, so a couple more things, and we will talk about divergent evolution a little bit later on. But in one of our targets, it says explain the relationship between changes in the environment and evolutionary changes in the population. So, this is an easy one. What evolution occurs when similar selective pressures result in similar what adaptations in different populations or species? So what do you think is going to fit in those two blanks? So you should have two words that you're going to respond to. What evolution and what adaptations? So get ready for our waterfall chat. You got this. You should have two terms in there. Okay, do you have it? Waterfall chat. Let's see, Mr. Akampora, what do we see? Any responses?
Just mine so far. Are people slow on the typing? But I said convergent because I looked at the next slide.
Yeah, that was a little bit of a giveaway, wasn't it? And what about the next?
I said successful adaptations, but I'm not sure that was the word you were looking for.
Yeah, in our learning targets, they preferred the word phenotypic. Adaptations in different populations or species. Now, this idea of convergent evolution, I don't have this in my slide, but I do want to bring this up. And there's some wonderful pictures of convergent evolution and that we could share from your textbooks or from a variety of sources. But in this slide, when we think of convergent evolution, do we think of common ancestry or not per se common ancestry when we think of convergent evolution? So I want you to think about that for a moment. So would the species have a common ancestor or not have a common ancestor? What do you think? And then we'll talk about structures, analogous structures versus homologous structures, just a little bit before we move on. So in your waterfall chat, let me ask you this question. Convergent evolution, common ancestor or uncommon ancestor? What do you think? Type in one or the other. Species with a common ancestor or not a common ancestor as an example of convergent evolution.
Ms. Bonzo, are you going to give us the 1, 2, 3, go?
Oh, waterfall chat. Waterfall chat. Sorry, Mr. Akampora.
We're playing Simon Says for a minute there. You didn't say, may I? Or, gosh, it's been a while since I played that.
Okay, so what are we getting?
Okay, we have a no comment and a comment and a non-comment. We seem to be split from the same people who are contributing. So all you other people out there, stop sitting on your keyboards. I mean, your hands.
Nice.
Don't sit on your keyboards either. But so it's split 50-50 right now, it looks like.
Okay. Yes. Uncommon, non-common. It would be correct. Good job. And I want you to think about how this applies to analogous structures. What are analogous structures? I know you've seen some awesome photos that your teachers have given you about homologous versus analogous structures. So when we think of analogous structures, we think of what? Examples like, can you give me one? Okay, let's type in our chat box, maybe an example of an analogous structure. Remember, they're not going to have a common ancestor per se. So an example of an analogous structure. I don't have this on my slide presentation, but it does apply. So waterfall chat. Any examples?
Wings seem to be the favorite one. I said bird and insect wings, and then we had Brennan shared wings of birds and bats, which seems like a much better example because I know bats are mammals, aren't they? I think.
Fabulous.
Birds.
Yes, fabulous.
That's mammals.
Let's think for a moment. Yes, I'm thinking that they are.
Okay, I thought so too, but then I couldn't remember if they gave live birth or had bad eggs. I guess I've never seen bad eggs. Remember going through caves and seeing lots of bats, but
Oh, yes.
I didn't see any bad eggs, so I guess they would have to be.
Yes. So excellent, students. So we know that analogous structures result from uncommon ancestry and that they can have similar functions, but not common ancestry. So
And that's because they evolve in response to the same environmental factors. Is that that's what we mean by convergent evolution, Ms. Bonzo?
Yes, sir.
Okay.
You are right on, Mr. Akampora. Thank you.
So bats and birds both had a need to fly, and I know that's a terrible way of saying that we don't evolve because we see a need for something, but because in bats and birds, flight happened to be an evolutionary advantage, those mutations that resulted in the ability to fly conferred a selective advantage. Would that be a good way of putting it?
I think that's a fabulous way of putting it.
I know I'm trying to fly myself, but I haven't been successful so far.
We wish that we could all fly.
Maybe our ancestors or our descendants, not our ancestors, that would be the other way. Our descendants, several generations will be able to fly.
Absolutely.
We all work on it.
It is wonderful having you join us today. Thank you so much.
I haven't joined myself and I'm learning some biology. I'm ready to take the AP Bio exam.
You're ready. You're ready. We're so ready. Okay. Explain how random occurrences affect the genetic makeup of a population. Okay. In review, I know you've had this before if you've had this unit. So I just have a few bulleted items about random mutation. I know when you had your genetics unit, you were studying a lot about mutation. We have this concepts of genetic drift in small populations. And in genetic drift, we primarily discuss bottlenecks and the founder effect. So I have a little picture here of an example of bottleneck effect. I don't think I have founder effect in here, pictorial, but we can talk about it. With genetic drift, this can cause big losses of genetic variation for small populations. Now, key really I'm finding with bottleneck and founder effect is small populations. So keep that in mind. Population bottlenecks occur when a population size is reduced for at least one generation. Okay, in this little pictorial, we can see the original population here, and then some kind of possible catastrophe could have occurred, something drastic to reduce the population, which created this bottleneck. So we have just a few survivors here. Here they're surviving individuals, and then of course the next generation. Now the founder effect, I always think about humans per se. I guess you could use this as an example of occurs when a new colony is started by a few members of the original population. We could think about how we came over from other continents, how we came from Europe over to America. I always think of that. It helps me to remember founders of the United States of America. So just keep in mind, those are pretty important in our understanding of what else drives evolution other than natural selection per se. Also, the last one, migration and gene flow, how this is just a little pictorial of immigration. You're exiting or leaving a population, immigration, you're coming into the population. And I love these little organisms. They have their alleles on there. Isn't that wonderful? Showing their homologous recessive traits. I just think it's beautiful. I love biology. So I'm so excited that Mr. Akinpour asked me to share with you today. Okay, couple more things and we're ready to do some practice questions. Here we go. Describe the role of random processes in the evolution of specific populations. If genetic variation is reduced within a given population, the differences can what between populations of the same species? I want you to think about that for just a moment. And what do you think? What do you think? So, waterfall chat.
We have decrease, separate. I'm stumped on this one.
Yeah.
And perhaps I think, Ms. Bonzo, I'm not exactly sure how you're using, well, how the words population and species and what the relationship is among those. Would you maybe explain that a little bit first?
Yes. Okay. So let's break this apart a little bit. If genetic variation is reduced, okay, so we have a population of the same species. So species means what? What does that mean when you're thinking of an organism that they can do what? They can breed and they can reproduce viable offspring. Okay, so they're within or they're in that population. So if genetic variation in that population is reduced within the given population, the differences can actually increase between populations of this species. So we would want to think of this as an increase between populations of the same species. I felt that was the most tricky when I went back and looked at your learning objectives. I felt that was a little tricky question there, but one that we could consider and think about. So increase would be an appropriate response. So describe the change in genetic makeup of a population over time. So let's think about that. And we might try this one as a waterfall chat. Okay, what results in genetic variation? We've mentioned this before already in our thinking, which provides phenotypes on which what selection acts. Two things we've already talked about. What results in genetic variation and what kind of selection? Really, we've only talked about two so far. One had 3 different kinds. Remember that when we talked about the squirrels and yes. So what do you think? Waterfall chat, let's go.
And we've got mutations and natural and mutations and random.
Nice, good job. Okay, we should have what? It should be mutation. Excellent. Mutation results in genetic variation, which provides phenotypes on which natural selection acts. Awesome. Great job, kiddos. Okay, now we are going to launch a poll. And I'm going to scroll down my page and hopefully We might need to share this document again if we've had newcomers come on board so that you can see the questions. I'm going to scroll down to some awesome AP Bio questions before we go into Hardy-Weinberg.
I put the link to those documents in the chat box again.
You are fabulous. Thank you, Mr. Akampora. Okay, so I'm scrolling. I hope I don't make you dizzy. Oh, my goodness, Miss Bonzo. All right, but this is so much fun. This is so much fun. All right, so we have 4 questions that we will launch a poll on. 1, 2, 3, 4. Because this is a cluster, because we just had a review, now we're going to apply our learning a little bit. And let's look at these questions. So 1, 2, 3, and 4, we will give you 6 minutes to work on these questions. That's 1.5 minutes per question. And I'm seeing that time is, how much time do I have remaining, Mr. Akampora?
Oh, 15 or 20 minutes. We'll have a little break between sessions, Ms. Bonzo.
Okay, I want to get to Hardy-Weinberg and the phylogenetic trees. So, all right, so let's go ahead and do this. And then I'll move into a Hardy-Weinberg. All right, so can everyone see this question? I want you to go ahead and answer question 1, 2, 3, and 4. It's multiple choice. I will read the question and then we will launch question number 1. You will put in an answer. And then I'll ask Mr. Akampora if he can launch that for us. So question number 1, and here's the scenario. A biologist spent many years researching the rate of evolutionary change in the finch populations of a group of islands. It was determined that the average beak size, both length and mass of finches in a certain population increased dramatically during an intense drought between 1981 and 1987. During the drought, there was a reduction in the number of plants producing thin-walled seeds. So I will be quiet so that you can answer 1, 2, 3, and 4. And then I will scroll if you don't have this document open. So take just a few minutes, please.
And students, just please make a note or write down your choices, and then we'll launch the poll separately. We can only launch one poll at a time, Ms. Bonzo.
Thank you, sir. Okay, I know I didn't give you 6 minutes, but let's go ahead and launch our poll for question number 1. Let's talk about question number 1. So if you could respond, 100%, we have, was it 1 response so far? So please.
And it's unfair. It's not letting me vote, Ms. Bonzo.
Ah,
I'm technically host.
it's just wonderful to have you.
We only have 3 of our 10 participants responding, so let's go, guys, please.
Okay, we have more coming along there. Awesome.
Should have the answers already, just have to type in the letter. All right, so that's a minute. Let's go ahead and end the polling
Let's go
here.
ahead. All right, so we had 100% absolutely correct. Okay, let's move on. That was the correct response, so we won't discuss that any longer. Let's look at number 2. Let's relaunch for number 2.
I'm doing that now. These choices are anonymous. I don't think anyone's grading the students on this.
No, I can't even see who is selecting what response.
I can't either.
And the total, like we can see the total poll. We don't know
Mhm.
you're teasing. So you're doing good.
Okay.
guys. Okay,
So-
so I'm going to. All right, it looks like, oops, share results, pardon me. Okay, so we have 4 that chose A and 1 that chose C. The answer is A is correct. Let's talk about maybe C. This option is incorrect. There are no data to support this conclusion that draws a correlation between beak size and flight muscle strength. Okay, so that's our rationale for not choosing C, but that A would be correct. Okay, so let's try number 3. And number 3. Oops. Think we're okay.
It says you've closed the polling, so
Oh, sorry. Okay, let me try again. Pardon me, kiddos.
if you voted, please go ahead and vote again on number 3.
Yes, please.
You know, I saw some responses I would agree with, and I hope that means they're the right responses.
Yes, it's correct. I think these kiddos know natural selection really well.
Well, better than me. I'm going to confess that I think I missed that first one.
Okay.
Okay.
All right. So we're on number 3.
This is number 3.
He is correct. And I know we have a couple of responses for C. So let's talk about C. Here's the rationale for not selecting C. The data provide no evidence to support the conclusion that new alleles appeared in the finch population through mutation. While within the population, there was variation in beak size and changes in the environment, which would be drought, provided a selective pressure favoring finches with longer beaks to crack thick-walled seeds. Okay. All right. So let's see. We have one more. Let's launch last question. You guys, you rocking this out on natural selection. Good job. Oh, I'm sorry, Mr. Akampora.
Did you?
Or did I do it again? Number 4.
I think you did. Yes.
Pardon me.
Okay.
I think I'm just, will you do that, Mr. Akampora? You want-
Well, that's either way. We're fine. Just please vote again. Or
Thank you.
it's not really a vote because we're not going to, person with the choice with the most votes isn't necessarily correct, although in this case they are, only because you guys are doing a good job.
They are
Like,
fabulous.
yeah,
Okay.
I hate saying voting as if we're selecting a prom queen or something.
You are fabulous. Okay, there are two concepts that are really important that I'm going to just go over briefly, and I'm so sad because our time is coming to a close. But a couple of questions. Stop sharing. Let me exit. Let's see, we want to exit this poll.
So B was the correct answer. Is that correct?
Yes.
We should make sure we're, okay, good job, all of you who answered.
Yes, and these are practice AP biology questions, so you are in it to win it. It's a wonderful job. All right, my favorite. Oh, I'm just going to run out of time. Okay, so I'm going to go ahead and scroll down. I haven't given you a review of Hardy-Weinberg, but I know you've had it in your classrooms. It's just one of my favorite math. I just love word problems, and I just think it's fabulous. So I do want to go over this one problem. And then I'm thinking, Mr. Akampora, that I have to stop. But number 7, let me give you the 2 key problems I want you to work on. Number 7 is a Hardy-Weinberg problem. And then I'm going to scroll down. We may not get to this, but number 10, okay? 2 really important questions I want you to practice and review. I do not get to review them with you today. Number 7 and number 10, okay? Your teachers have the answer key for these 14 questions that are really important for your study on this unit. So let's go back to Hardy-Weinberg. When I do Hardy-Weinberg problems with my students, I tell them to write the alleles at the top of their problem as they are reading the problem. And that's very, very helpful. So I'm going to try to annotate just a little bit. I'm new, Zoom's not my big thing. I typically use Google Meet, but I'm going to give this a whirl. Okay. In a population of certain frogs in which the allele for brown skin is dominant. Okay, Hardy-Weinberg, dominant is P, powerful, always, okay? So think of it like that. To the allele for green skin, all right, so we know what dominant is. I'm just gonna use big B. It could be what? It could be big B, as brown skin, or it could be heterozygous, right? Okay, could be either one. To the allele for green skin, a drought leads to selection against green skin frogs. So green skin, we know, will be represented as what? Little b. So this is what I tell my kids. Read the problem and annotate along the way. A drought leads to selection against green skin frogs. When the drought ends, 12% of the remaining frogs exhibit green skin phenotype. So 12% are going to have that homozygous recessive little b phenotype. If the population is now in Hardy-Weinberg equilibrium, what will be the frequency, this is important, frequency of the green skin allele in the next generation? Provide your answer to the nearest 100th. Okay, so my kids focus on P plus Q equals 1. Okay, focus on that. All right, then all you have to really know is the inverse math operation from going back and forth from being able to use your square root function. Truly, that's all you need to know, pretty much. So what are we going to solve for? We're solving for what? We're giving the percent in the total population. So we know that's 12%. So we're going to make that a decimal so we can think of this in terms of frequency. So 0.12 is the same for us as 12%. Now, this is going to represent what in our population? What allele? Is it going to be what? little b? Is it going to be big B, little b, or big B, big B? What's that kind of going to represent? Well, it's going to represent this. Okay. Now, we don't know. We do know that 88% of the population is combined this, right? Okay, we know that. So how can we say this is going to represent here? So how can we find Q if we know that this is Q squared? How can we find that? We have to take what? The square root of it. So when we take the square root, someone tell me what this is in the waterfall chat. I'm so excited for you to give me the answer and the response if you know it. Are we ready? You had your handy dandy calculators ready to go. And the answer is what? Waterfall chat. We could launch a poll too. Do they have an answer, Mr.
We
Eccles?
said A. I was getting my calculator out to find out the number, the actual number, but our choices seem to be A.
Yes,
I
sir.
got
That's correct.
34641, but that's
Yes, sir. Yes, sir. Excellent job, kiddos. And again, please, I'm going to be saying goodbye because it's time for me to end the session, but please make sure you review question number 10 in your studies, and your teacher has the correct response and answers for you for all of these practice questions 1 through 14. Thank you, Mr. Akampor. I think my time is over.
Thank you, Ms. Bonzo. That was, I know I enjoyed that, and I feel ready to take the AP biology exam. Students, we're going to take about a 5-minute break and give you a chance to use the restroom or get a little snack, and then we'll join Ms. Cook, who will be helping us learn about or review ecosystems and ecology. So now, and welcome back, students. Ready for our second hour on ecology, and let me introduce Ms. Cook to take it away. So take it over, Ms. Cook.
Hey guys, so today my goal for you was to spend more time on free responses and kind of tying in a lot of content from the year with ecology. I kind of feel like ecology is one of those units that concepts are big and usually come much more easily to students. So we'll work on some free responses, and hopefully we have some time to do some ecology math as well. So let's see. All right, so the first thing that I have is just to kind of talk about where I want to go. And so keep in mind that organisms have to respond to their environment, and the reason for that is it allows them to grow, it allows them to reproduce, and it helps them maintain this balance within them. And so all of that depends on them responding appropriately. And so I'm going to start with plants. I think for me, that's just something that I'm not the most familiar with. And so when it comes up on test questions, I would want you to have access to these two topics. So phototropism and photoperiodism, these are common. I think you can see data, experiments, and have questions on that. So we'll start with phototropism first. So phototropism is where we have a plant growing in the response to light. And so we can have negative phototropism. And so that would be the root of the plant, obviously, grows away from the light. And positive, the shoot of the plant grows towards the light. And so auxin is going to be a hormone that's responsible for positive phototropism. So you have this chemical molecule that's going to respond to an external cue. So this light and then chemical compound would help facilitate a response in the plant. And I had never seen a picture like this, kind of going into the details of how a plant grows towards the light, but I thought it was a great picture to show you a lot of the content that we've covered this year. And so if you look at this picture, if this was something that came up on your test, hopefully you would see this picture. It has a cell membrane and it has a cell wall. So hopefully you would be like, that's a plant cell. And so this center structure here, it's really big. I'd like you to go ahead and tell me in chat. So this takes us back to Unit 2, knowing your cell components. What do you think that big structure is there? So in chat, if you'll go ahead and type in your answer. What do you think this big, large structure in the middle of a plant cell is?
We may be waiting for a cue, Ms. Cook.
Yeah, and you guys can go ahead and respond. So
Let's
we
go
can,
ahead and type when ready. And I have no idea. I'm not a botanist. I can't help you here. I feel so ignorant.
this is one of the 3 structures that plant cells have that animals do not.
Oh, okay. Well, now I feel a little bit better.
Okay.
We've got a couple of good students saying vacuole and central vacuole. So
And that is perfect. And so kind of keep that, things that are familiar to you, let those kind of guide your thinking. And so in this next picture, we're seeing that auxin is stimulating this protein, which is specifically a hydrogen pump. This might be called a proton pump. And so hydrogens are going to go across the membrane into the cell wall. And so my next question to you would be, what do you think is going to happen if we have a high concentration? Or what is the change if you have a high concentration of hydrogen ions somewhere? I'm going to see if you remember. So you can go ahead and type whatever you're thinking. So if you have a buildup of hydrogen ions, so that's going to happen on the cell wall side. What's going to happen? What will be a change? So chemistry is coming into play here. Do you all agree?
I'm muted. You're in my wheelhouse now.
All right.
I
So
don't know what effect that has on the proteins or other things in there. Now I've got to defer to my biology friends.
so it does lower the pH. And so if it has a low pH, remember that's acidic. And that's weird. That's the weird thing about the pH scale. I think that's hard. So when your pH numbers go down towards 0, you have a higher concentration of those hydrogen ions. And so that gets confusing when we think about that. And so if our cell wall becomes acidic, remember that's going to affect the shape of molecules. And we probably spend more time, I know I'm guilty of this, I spend more time talking about the effect on proteins. But your cell wall is composed of, if you kind of look at this little picture, cellulose. So that's that polysaccharide that kind of helps give that rigidity to the cell wall. And so if it becomes more acidic, it loosens those cellulose fibers. And now we have some flexibility to elongate the cell. And that's what you're seeing over here in this picture, that the cell wall elongates, water moves in, and that's going to help fill up that central vacuole and helps elongate that cell so it can actually move towards the light and bend towards the light. So here's another question. So see if you guys remember, if water moves in from the environment, so water moves into this plant cell, it fills up this central vacuole. There's a term that we give plant cells. They kind of fill up and they become kind of nice and full. That central vacuole is going to push against the cell wall. Do you remember the term that we give plant cells? And again, you can go ahead and go in and type. Okay, so I am seeing hypotonic. They are in a hypotonic. If water's moving in, there's my word. So you do know biology, Lou. So turgid. Turgid would be my word. It's kind of nice and full for a plant cell. All right, so I did like that picture. I've never used it. I found it when I was kind of looking at phototropism, but I thought it was a great example of a lot of things that we've talked about. All right, I'm not going to go into a lot of details. So as far as phototropism goes, there have been several experiments. So Darwin, that you're familiar with evolution, and his son did experiments, these first 4 things that you're seeing. And then Boyson and Jensen also did their own experiment. And so I kind of like this picture too, based on what we had just looked at. Remember, the light side, or the side of a plant that's getting the light, isn't the side where the plant cells extend or elongate. It's actually the shaded side. So I just wanted to show you this. This is experiments that have been done, and I wanted you to see what AP College Board did to make a test question. And so I want you guys to look at this, very similar. I think most of it's, they took out a few of those examples that Darwin did. So you were given that phototropism in plants is a response in which a plant shoot grows towards a light source. So the results of 5 different experimental treatments from classic investigations are shown above. And so for part A, they want you to give support for the claim that the cells located in the tip of the plant shoot detect the light by comparing the results from treatment group 1 with the results from group 2 and group 3. So let's start in chat, just talking about comparing group 2 to group 1. So your job is to tell me or give me support for the claim that the cells located in the tip of the plant are what's detecting the light. So compare 2 to 1 in chat first. So how is group 2 different from group 1? You can go ahead and show your answers. I know typing takes a little bit longer.
Okay, it's just, this is a little bit of typing for us here, Ms. Cook.
I know. Let's see. Okay, so it looks like here's how they gave the point. So in treatment 2, the tip's removed and the plant no longer bends towards the light. So that would be the only thing. Just make sure that if you're saying the tip was removed, make that connection that it's also not bending towards the light, and that's how they're different. All right, so the second part of that is they also wanted you to compare treatment group 3 to 1. So again, the claim that you're trying to support is that cells in the tip of the plant detect the light. So what's going on with 3 compared to 1? All right, does anybody else want to put your answer in? So Brennan kind of answered 2 and 3 together. That's fine.
Mhm.
All right. All right, good. So here, what you would want to notice is the opaque cap. And so if you didn't really know what opaque was initially, at least look at the plant. And so it did grow straight. So obviously the tip of the plant was not able to receive the light. So therefore it didn't bend. So if you mentioned that the cap blocked the light at the tip, then you would have earned the point. All right. So for B, in treatment groups 4 and 5, the tips of plants are removed and they're placed back onto the shoot on either a permeable or impermeable barrier. Using the results from treatment groups 4 and 5, describe 2 additional characteristics of the phototropism response. And so the thing that I want to stress here for test A is they're asking for 2 things. And they do give a point for each one. Here's the thing you don't want to do. So if you think of 2 things and then you're like, I'm not too sure if one of those is correct, I'll put a third one. You're just wasting your time. So we still read those. We have to read everything. But if one of your first 2 is incorrect, we don't give you credit for the third one. So just do 2, be confident, and move on. So we have a permeable and impermeable barrier. And they want you to describe 2 additional characteristics of phototropism. So what do you think you would say? What else is going on at the tip of the plant? So I'll give you about a minute to throw at least one in there. All right, any last answers? All right, so you all are on the right track. So you're seeing that something is occurring through the permeable barrier, but not the impermeable. And so what they wanted you to pick up on is that the tip, since it's receiving the light, that had to be present. The tip is producing a chemical. You could have said a substance. If you had studied this particularly in class and knew that it was auxin was the hormone, that was causing the plants to bend. And then the second thing that you would have gotten a point for, if you said that substance then diffused through the stem, so from the tip to the stem to cause that bending, you would have earned a second point. But that was what they wanted you to pick up on with that one. All right, so let's move on. So I do have answers here if you want to go back and look. So I have this there as well. All right, so I'm showing you this picture to set us up for the next slide. So this is a Phytochrome. It has photoreceptor activity, so it can receive light, but it also has kinase activity. And so this takes us back to Unit 4 when we talked about cell signaling pathways. Do you guys remember what the job is of a kinase? So go to chat. Tell me what a kinase does.
I think you've got me stumped here, Ms. Cook. I'm going to take a guess.
It is going to help relay signals. But it has a specific job to relay the signal.
Oh no, my chat's frozen.
I know mine was kind of slow to get access to again. It usually is involved in the step of amplification. Good. Any other last thoughts? So kinase's job usually in the transduction step is to add a phosphate group to another molecule. And so that changes its shape, and that kind of helps send the message on until we can finally get a response of the cell. So what I've given a little bit of background before I move to the next slide. So when a seed germinates and reaches light, it's going to go through something called de-etiolation, or this greening process. And so the stem elongation will slow, the leaves expand, roots elongate, and chlorophyll is actually produced by the shoot. And so you have this pathway showing you that process. So again, I wanted to tie in Unit 4 to how a plant can respond to external cues appropriately. And so what we're looking at here, there's your little phytochrome I just mentioned. So light comes in, hits that, and then that causes it to change shape. And that's going to activate that kinase part. And so you guys, one of you said this, you said it helps in amplification. So what step of a signal transduction pathway helps in amplifying a signal? So is it step 1, I'm referring up here, step 2, or step 3? And so you can type A, B, or C, or the steps, whichever one, into chat. So kinases, what step of the pathway? My chat's being slow. All right, good. So B, or step 2, is the transduction, is where we're going to get amplification. And so once that phytochrome actually detects the light, it changes shape, and then it's going to phosphorylate. So these arrows, they're trying to simplify this picture. There probably is a phosphorylation cascade here. We activate a second messenger, and then we finally activate a specific protein kinase. And if we look, we can kind of see its job of phosphorylation. So we have a transcription factor. It becomes phosphorylated thanks to this kinase. Its shape is different. And so now it can dock to your gene and start transcription and translation so that we get these greening proteins produced. Same thing over here. So the phytochrome can activate a calcium channel. So here's a good tie-in to transport. What kind of transport is occurring to bring the calcium ions in to this cell? So again, go to chat. What type of transport brings those calcium ions in? Oops. All right, so we've got facilitated diffusion or diffusion. And so the key here is you have this protein channel. Calcium ions are charged, and because of that, they're not going to be able to get through those fatty acid tails of the membrane. And so they're going to use the help of a protein to facilitate. So facilitated diffusion is what you'd want to go with. So it too is finally going to activate a kinase. And I just wanted to point out, last week, if you were with me, we talked about gene regulation, and kind of think about this. If a plant is just a seed, or it's maybe grown a little bit, but it hasn't reached light, then there's no point of it wasting all these resources and all the energy to produce these proteins that allows it to green if it's not even, at the point of receiving light. So all this is not going to be going on in a seed or as it kind of grows through the ground, but as soon as it's able to receive that light, then it wants to turn on these genes and express those proteins so that it can grow and maintain homeostasis and respond to its environment appropriately. All right, so let me check time. We'll go ahead and talk about this one. This should be quick with a multiple choice question. So photoperiodism is another way that I think they could question you on plants responding to environmental cues. And so photoperiodism is a plant's response to a photoperiod, so the length of night and day. And they really figured out through experiments that nighttime or darkness was really the critical factor. And so I've got a picture for you. Short day plants or long nights. So long nights really the key here. They prefer longer night. And you're seeing a dashed line in the figure that shows the critical night length. And so in this first one, if the light kind of exceeds that time, so there's not as much darkness, then the plant doesn't flower. But in this one, we see that the darkness is longer and the light time is shorter, and so they do flower. And then even in this one, if there's kind of a brief flash of light that breaks up that dark period, those plants will not flower. Long days are just the opposite. They kind of depend on a short night, and so we kind of see the results here. And so here's a question. If a long day plant has a critical length, and so remember that's based on darkness of 9 hours, which 24-hour cycle would prevent flowering? And so you can just do A, B, C, or D in chat. So again, the key is remembering that critical length is 9 hours of darkness. And we want to know what would prevent flowering. Anybody else? A, B, C, or D? All right, good job. So B is the one. So 10 dark hours, that would be too much. And that would prevent flowering for the plant. All right, so moving on to animals, I think this is what's most fun, and this is a good tie-in to natural selection and evolution. There's lots of ways that animals can communicate, to work together, or a lot of times I think we talk about this most in biology is sexual selection, when males are trying to get the female's attention most often. And so I just wanted to kind of give you some ways. I feel like most of these terms are familiar to you, but I did want to stress, and I won't take our time, tactile. If you haven't heard that, that's touching. A lot of times bees will do that to tell each other where food sources are. And then I wanted to mention olfactory. I feel like that's come up in question stems, and I don't feel like I use that term very often in my class. And so that would be the sense of smell. So I did want to just put those words in front of you before test day, just in case you hadn't talked about them. But a lot of times animals will also communicate to, show dominance visually. Males that are really big, they're usually the dominant males in a group. I mentioned tactile to help them find food. If I walk my dog and she, pees quite often, she's trying to mark her territory. Cats do that as well. And so there's so many things that they can talk about. Hopefully you guys had a chance to do like the pill bug lab or behavior lab. Those can be a lot of fun. The other thing that I want to talk about, natural selection. So not only does it help them in mating successfully, but there's innate and learned behaviors that would help them survive and reproduce. And so innate, if you're seeing that word, they are born with that characteristic or that trait. And so a really good example is suckling of a baby. So we don't have to teach them, and that allows them to survive and know how to get their food just from the very beginning. And then learned behavior is based on experience. And so one of the, a good example I think is dogs are curious. They come into contact with a porcupine, and that's probably the last time they're going to be curious about a porcupine in the future if they get those needles in its nose and that kind of thing. So there's lots of fun experiments that have been done, and you guys can probably think about this with your pets. But learned behaviors, there's something called associative learning. And so it's where animals associate one thing to something else. And so I'll give you an example. I love talking about my pets. And so my dog loves to walk, and we keep her leash in a drawer. And every time you open the drawer, the handles kind of clank. And so every time she hears that clanking of the handles, she comes flying through the house thinking she's going to go on a walk. So that's a perfect example. Both my pets, I have a cat and a dog, if they hear a bag rattle, they think that's treats for them rather than anything else. So, and you guys are probably familiar with a lot of those as well. All right, so the next one, I think some of these questions, especially the kin selection, I think came up on, and maybe just altruism in general, I can't remember, was on your mock as far as the multiple choice. And I do think this is an important and maybe more often asked question on the AP exams. And so I do want to mention again, so altruism is something where a species will warn others. So they kind of put themselves at risk to save others in the population. And this ties into evolution pretty well. So my example, I witnessed this the other day, and I'm going to have to do some research, but I was sitting in my backyard and I have a bird feeder. And so I was sitting with my dog, not really paying attention to too much. I had seen my cat. So this is my cat, Lucy. She loves to kind of walk down the edge of my fence, and she's behind flowers. So she gets to hide and watch the birds come to the bird feeder. And so she's gotten to the point where she grabs the birds and brings them into my house, which has not been fun. But the other day we were sitting in the yard. She was hiding. I kind of forgotten about her. There was a blue jay on the fence, and I was busy watching this other little bird. I'm not sure what kind it was, but it was kind of trying to come to the bird feeder, but it wasn't sure if it should. So I was watching it, and then I hear this horrendous screeching, about 4 loud screeches coming from the blue jay. And I look over, and so he had spotted my cat hiding and had sent out that call to warn the other birds. And I thought that was interesting. I didn't realize they would warn other species of birds. So I thought that was kind of fun example. So on the other hand, there is a specific type of altruism called kin selection. And so meerkats are super cute, and they're a great example of this. So if meerkats are out eating, and if one of them spots a predator, they will give out the warning call with the idea that even if they are killed by that predator, their family members will survive. And so family members have genes in common. And so the reproductive fitness of that particular organism, that particular meerkat that gave the warning call, can still be passed on. And so Haldane, it's JBS or JDS, I can't remember his initials. He has a saying that he would give his life for 2 brothers or 8 cousins. And so the idea is that your siblings have half the genetic information as you. So it would take 2 siblings to equal you as an individual, passing on your genetics, or 8 cousins. So 8 cousins are less closely related. So they have about 1/4 of the genetics in common to you. So if you had 8 cousins live because you gave the warning call, then that would essentially equal you still living and passing on your genetic makeup. All right. So I wanted to give you this free response, moving back to plants and how they can protect themselves. So we kind of looked at how animals communicate and can protect themselves from predators. I wanted to give you a way that plants can, but then tie in a lot of important skills that you're going to see on your free responses this coming Friday. So trichomes are hair-like outgrowths that protect plants from herbivores. And so they wanted to investigate variation in stem trichome density within a plant species. And so a student counted the number of trichomes on the stems of 6 plants from 3 different populations. So the student used the data to calculate the mean trichome density for each population, and the results are provided in this table below. So for part A, you were asked to create an appropriately labeled graph to illustrate the sample means. And so those were calculated for you of the 3 populations to within 95% confidence. So if you see this, that's the same as plus or minus 2 standard error of the mean. And the reason that I wanted to give you this one is you have to pay attention to what they're providing. So in the data table, we only have SEM, which is the same thing if they have plus or minus 1 SEM. They want us to graph plus or minus 2. So all the SEMs are 1. So in chat, could you tell me what plus or minus 2 SEM would be? So if 1 plus or minus 1 standard error of the mean is 1, what would plus or minus 2 standard error of the mean be? It's actually pretty easy. All right, so Lou has put in an answer. Do you guys agree with him? He is a chemist. Can we trust him?
I know, it's all those small molecules that are so easy to deal with compared to your big molecules.
All right, so it really is that simple. And I wanted to show you this one in case they tried to trick you, but please don't take me like you have to multiply every time. So my students did that. So only you have to look, are they giving you plus or minus 1 standard error of the mean and they want you to plot 2? Or did they give you the 2? Then you would just plot the numbers that they provided. So if this had said plus or minus 2 SEM in the data table, then you would just go up and down 1 from the mean, okay? So be careful. I just wanted to give you this one. I've seen this happen. This is probably the only question that I've seen them do this before. So they may do it again. So instead of graphing, I kind of wanted to show you 2 graphs of student responses and see which ones you would go with or how you would give points. So I'm going to move to the next slide for a second. So here's the data again. Here's the means, 9, 11, and 14, and the standard error was 1. But remember, they wanted us to plot 2. So plus 2 and minus 2 from the mean. And so the points that they gave, there were 3 points for the graph. So if students labeled, scaled, and had proper units, they got a point. If it was a bar graph or a modified bar graph, which is this, with appropriately plotted means, they got a point. And then if they had 2 times standard error above and below, they earned a point. So let's start with this graph on the left. How many points do you think this student earned for their graph? So 1, 2, or 3. You can throw that in chat and we'll try to move on to the next part. All right, Abby's agreeing with Lou, 3 points. All right, yeah, that one got a 3. So one on the right, how many points do you think it earned? 1, 2, or 3? All right, so you guys are saying 1. And let me make sure before I say wrong. Yeah, so they got 1 point for they correctly plotted the means. So that was the point that they earned. Good job.
If Ms. Cook, I think I would have given them a second point had they labeled instead of mean trichromes per square centimeter.
Yes.
So is that something that is important to make sure you're labeling your axes correctly or within
Right,
the
in the
information?
units, yes, definitely. And that's the thing, like I would also say is, you never know, I always encourage my students to always give a title too, because you never know how they're going to incorporate, like this one, you had to correctly label, scale, and have proper units just to get 1 point. Well, for your test this year, they might throw in title. You have to have a title too to get 1 point. So you need to always think about all those components every single time. Here's the other thing that I'm part of it. I've never had to, grade a graph on the real AP test, but I'm part of an AP group in Facebook, and those teachers recommend that you do your graphs in pencil so that if you do make a mistake, you have a chance to correct it because, pen is definite. And then if you have time at the end of your test, go over it in pen so that, nothing smears or whatever. So do a graph in pencil so that you have the chance to fix mistakes, and go over in pen if you have the time at the end. So I would make that suggestion. I think that's a good one. All right. Oh, the other thing I would tell you, if you are doing error bars, don't shade in the bar that's representing your mean, because they're not necessarily going to, just make sure that they're going to see your error bars extend down. So usually you don't want to shade in on that one. All right, so I'm going to go backwards. So based on the sample means and standard errors of the means, identify the 2 populations that are most likely to have statistically significant differences in the mean stem trichome density. And I'm going to use that graph that got all 3 points, and then you had to justify your response. So you're just identifying here the 2 populations that are most likely to have statistically significant differences, and then justify why you're saying that. So there's the graph, the one on the left. Use it. Which 2 populations are more likely to have statistically significant differences? And then why are you saying that? So again, your job is to identify the 2 populations, 1, 2, or 3, that have statistically significant differences in the mean stem trichome densities, and then justify why you chose those 2. All right, and so this goes along with interpreting these error bars. So if you are making a graph, those error bars are really important. And the way I'm interpreting the new, we have a new test, kind of a new style of test. And so I feel like you are definitely going to have to plot error bars on question number 2 for sure. So being able to interpret them is important. So 1 and 3 is correct. And so why are we choosing 1 and 3? These error bars do not overlap. And so that means that the data, since they don't share any data in common, that there is a statistically significant difference. So if I compare population 1 with 2, their error bars overlap quite a bit. So there is not a statistically significant difference between those 2 populations, and same with 2 with 3. Those error bars overlap. And so that data, they share data, so there's not a statistically significant difference. All right, last part, and I like this part because it gets all the parts of an experiment in. Oops, before I give you the answer away. All right, so for part 3, we are going to describe the independent and dependent variables and a control for an experiment to test the hypothesis that higher trichome density in plants is selected for in the presence of herbivores. My suggestion for if you ever have to support a hypothesis or support a claim, make sure your focus remains on the hypothesis or claim. So the hypothesis is higher trichome density in plants is selected for in the presence of herbivores. So in my opinion, when I tell students to write hypothesis, it should always include the independent and dependent variable. And so if I look at this hypothesis closely, higher trichome density in plants is selected for in the presence of herbivores. Which one of these things am I going to be measuring if I do this experiment? So if I'm measuring, that's my dependent variable. So higher trichome density or presence of herbivores, which one of these things will I measure? So go ahead, that would be your dependent variable. So go ahead and put in chat. Which one of those things do you think you would be measuring if you were doing this experiment?
And Ms. Cook, as people are typing here, I've seen in biology questions, they ask for a null hypothesis. And this is something I've never seen in a chemistry or a physics AP question. So perhaps you could say just a few words on what they mean by that.
Right, so.
Or give an example.
Yeah, so like for this one, the hypothesis is that higher trichome density in plants is selected for in the presence of herbivores. So a good null hypothesis would be that there would be no difference in trichome density in the presence of herbivores or in the absence of herbivores. So there's no difference, but I'm being very specific on what the situation's about, and I'm still including the independent and dependent variables. So, and I think that's the thing, my students did, and we saw with the mock exams, is just not the specificity. So there would be no, you could be no statistically significant difference in trichome density when herbivores are present or absent.
It always seems like a double negative when you're asked, we're trying to disprove, or no, we're trying, but the null hypothesis is disproven.
I know. Yes.
I have to really very carefully wrap my head around that.
Yes. All right. So the thing that I'm going to measure is how many trichomes per whatever centimeter squared, I think is what they were doing. What's the trichome density? That's what I'm going to measure. And so that's going to be my dependent variable. And then what am I going to change? What can I manipulate easily? So I can't make a plant produce a certain number of trichomes, but I can change this to see how the plants react. And so I'm kind of giving that away. Trichome density is my dependent variable. So the presence of herbivores would be my independent variable. And so again, my suggestion, really read that hypothesis, because, sometimes I think you think of an experiment and you're like, oh my gosh, there's a million things I could do. And maybe you've never heard of trichomes before and you don't know where to start. So really read that hypothesis, see if you can pick out the part that you're going to measure and the part that you have control of as an experimenter to decide the independent variable. And so that got you 2 points. If you could decide that correctly, trichome density is the dependent, presence of herbivores is my independent, and we've got one last part. So what would be our control group in this situation? So go to chat, see if you can type in what our control would be in this particular experiment. Okay, so types of plants used would be a constant. That would be something we want to keep constant. So what's our control? This is kind of, control should be opposite of your experimental group or the group that receives the independent variable. All right, any other answers? Okay, yes, so no herbivores or, no herbivores present would be our control group. Okay, so there was 3 points. The last part of this question is to identify an appropriate duration of the experiment to ensure that natural selection. So now your last study session, what would be an appropriate length of time to know if natural selection is favoring plants with higher trichome density? So how much time or appropriate duration to ensure natural selection is measured? So you guys can go ahead and type your answers in. How much time would it take to know if natural selection was actually giving us higher trichome density in plants? All right, so their answer, their duration, you would want more than one generation, right? You would want them to reproduce and see, how many trichomes they had. So with natural selection, if this comes up on an experiment, multiple generations would be needed to really know if there was a selection going on. All right, and then the last one is predict the experimental results that would support the hypothesis. So again, go back and read your hypothesis that higher trichome density in plants is selected for in the presence of herbivores. So what results would we need to get to support that hypothesis? All right, very good. So we're going to see more plants with higher trichome density in the presence of herbivores. Very good. Expect these types of questions. So A and B, you should be seeing on question number 2 on test day. C is possible, parts of that, but definitely question number 1, and I think it's question number 4 pulls in, maybe you have to make a null hypothesis or identify independent and dependent variables. So these are all good things to practice prior to test day. All right, how much time have we got?
Oh, maybe 5 minutes if you'd like, Ms. Cook.
Okay. So energy pyramids, I think the big thing I would want you to keep your mind on if you have something like this, I think we're bad at, as humans, we always feel like we're the top dog of the world and, you know, nothing affects us. But in the real world, if one organism is affected, then it is a trickle down effect to those other populations. So I would want you to always keep that in your mind, whether you're talking about a food web, a food chain, an energy pyramid. If something happens to one of those organisms, somebody else is going to be affected. And you can't be vague on free response questions. So you want to be specific. So is there an increase or a decrease? You can't just say they would be affected. So give direction there. So like in this case, what if amount of light decreases? What would you say is going to happen to these organisms in the energy pyramid? So again, you can type your answer in chat. If the light were to decrease, what are we going to see happen to this energy pyramid? All right. Good. So you're telling me a specific effectiveness. So it's going to decrease everybody. So producers for sure, but then if there's less producers, then there's less rabbits, there's less snakes, and it goes on. So just to make sure you kind of know the rule here, if 200,000 kilojoules of energy is available at the producer level, how much energy would be available to the snakes there in the secondary consumer level? And so this is some easy math that they could throw in there. It's not on the equation sheet, but sometimes they do that. So 200,000 kilojoules of energy at producer, how much at secondary consumers? All right, anybody else? So we got 2 at 2,000, and that is correct.
So it's 10% gets
Yes.
passed on,
10% at each one.
90% gets dissipated to the environment.
Right, and it's lost in the form of, can anybody tell me? A lot of energy gets lost in this form.
The inexorable triumph of the second law of thermodynamics. So confused, inclined.
All right, so Lou is correct. Heat is how energy is lost. I want to show you, so we are running out of time. I did try to tie in different equations that you may see. I'll be honest, I haven't seen these come up too often on the AP test in previous years. It definitely does not mean that's going to happen this year. So I feel like anytime I'm like, oh, that's never happened before, it happens that year. So I've tried to give you an example: population growth, exponential growth for you to practice, logistic growth, and then I've got an actual AP multiple choice question. The answers, if you go to Advanced Kentucky, the link where resources are and the videos are recorded, the answers are in the speaker comments at the bottom of the slides. And then I've given you some information about Simpson's diversity index. This is a new equation for our equation sheet this year. Yeah, there's the equation. So let me see what else I got. And then I did just kind of mention biodiversity. Keystone species was a multiple choice question on your all's mock test. So they were specific. I hadn't felt like I'd seen that in a couple years. So there's an example. And then of course, we always have to think of disruptions to ecosystems. And then I've got some mutualist or symbiotic relationships. This was one of your free response questions, was on parasitism. So I've given you some different examples there to think about. So I guess I'm out of time, but best of luck as you guys finish up your last week before test day. Think positive. I think that's the key to being successful. And if a question frustrates you, take a deep breath or move on. One question is not worth any more than any other, so don't let it eat up your time and knock down your confidence. So stay positive, and you guys are going to do super great.
Thank you so much, Ms. Cook and Ms. Bonzo, if you're still on. And thank you, students, for being here. Good luck on your AP Bio exams. I hope many of you are not graduating. If you are graduating, congratulations. If you're not, maybe we'll see you in another AP course next year. I think learning science, I enjoy learning, even at my advanced age, I enjoy learning a little bit of biology or physics or chemistry, and there's always more to learn. So those of you who are going to get on the computer, watch TV, it's a beautiful day out there. Go out and get some exercise, and then you can relax a little bit. But thank you so much, and I'm going to cut the recording and say goodbye. Thank you.
See ya.